Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or called CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Ann Stock. She's a professor in the Department of Biochemistry and Molecular Biology at the RWJ Medical School, and she's the associate director of CABM. Hi, Ann.
Hi, Marty. It's great to be joining you this afternoon.
I want to just get started by asking you, can you tell us a little about your education and your early career before you came to CABM?
I was an undergraduate at UC Berkeley, close to where I grew up, and I studied biochemistry there and started working in the laboratory of Daniel Koshland Jr., where I was one of many undergraduates working on bacterial chemotaxis. And I grew to love research so much in that senior year experience that I decided to go to graduate school. I stayed at UC Berkeley for graduate school, working in Dan Koshland's laboratory.
So what was your dissertation in graduate school?
It was on bacterial chemotaxis and the molecular basis of the signaling components in bacterial chemotaxis. And to some extent, I've continued to follow the same research path for the rest of my career.
Up to this day.
So I followed a spouse to Princeton University and completed my graduate years there from afar, still getting my degree from UC Berkeley, but taking a turn from biochemistry to molecular biology, still studying the same system. And then I began a couple of postdoctoral years there and had the wonderful opportunity of being required to extend my postdoctoral years when I received a Markey Fellowship. And so I had become much more interested in structural biology and what it could tell us about the molecular mechanisms of the system. So I headed up to Greg Petsko's lab in the Boston area. We'd been collaborating with him on a project, and I learned structural biology at Brandeis University with Greg Petsko for a few years before starting my position here at the Center for Advanced Biotechnology and Medicine.
Great. And so how long have you been here at CABM, and what was it that brought you here?
I've been here since 1991. And there were a couple of things that attracted me to CABM, mostly that it really had a wonderful focus on structural biology at the time. The center had been newly established, and I was really happy to be part of a multidisciplinary environment, which at the time was not really so available everywhere. Departments tended to be somewhat narrowly focused. And my research had crossed biochemistry, cellular studies, molecular biology, and genetic approaches, and then headed into structural biology. And so to come to a place that was combining all of these together into one center was really a fairly unique opportunity and a wonderful experience. And the center had been recently established and was attracting a lot of really young researchers who were very excited about what they were doing. And it was a fun place to start and become part of. In addition, I think I always wanted to end up at a large state school, having started at Berkeley and had a wonderful experience and an opportunity there. And I wanted to spend my career at a place where I could give back to that sort of a school.
Yeah, that's great. Well, it's now almost 30 years, so it seems to have worked.
Yes.
So let me ask you.
It's been a great environment.
So how about now? What would you say is the main focus of your lab? What are the key questions you're exploring?
Our main focus of research is bacterial signal transduction. Bacteria's single-celled organisms are exposed to an enormous amount of change in their environment, and they depend on signaling systems to be able to respond and adapt appropriately. So we've been interested through the years in a range of different levels of investigations, ranging from cellular down to molecular and atomic resolution detail of some of the proteins involved. But now we're back to a cellular level, really thinking about the principles behind the design of signaling systems. And we take a combined approach of experiments and modeling, where we study a model system in E. coli and define the parameters involved, then use computational modeling to see the types of behaviors that can come out of such a system, make predictions, and go back to the lab to do experiments to test out the models. And we're learning a lot about how sophisticated behaviors can come from very, very simple, schematically configured signaling systems.
Great, great. Now let me switch bases and ask you about one of your important functions, which is as a teacher. So can you tell us a little about how you interact with students, trainees, and other members of your lab?
Interacting with students and trainees is actually the highlight of having an academic position, and it's why I'm in an academic position as opposed to out in industry or elsewhere. It's fun to watch students develop, and it's really fun to watch them develop their own ideas. So in my laboratory, people have a lot of independence. They usually get started in one direction and then kind of develop their own independent projects and own independent directions based on their own particular interests. So many of the projects that are pursued in my lab have actually been really directed very much by the individuals themselves.
Great, great. So let me ask you, aside from your research at CABM, do you have other roles at Rutgers in the scientific community or the world at large that you want to share with us?
I participate actively in a number of different activities at Rutgers. One of the things that takes up a fair amount of my time is co-directing the graduate training program in biotechnology, which is a great program that brings together biomedical engineering students with students in the chemical and biological sciences for a lot of interdisciplinary interactions. And a lot of collaborative projects.
So now here's a difficult question. Imagine I'm writing your obituary, and how would you like to be remembered for your work?
That's actually a pretty easy question. I think that more than the work itself, I'd like to be remembered for the people that have come through my laboratory. Science is certainly a continuum where each little incremental bit of knowledge builds on what came before. And so it would be naive to think that there would be any discoveries that stand the complete test of time without evolution. And it's the people that are involved in the research that contribute to that. And so having lasting research really is accomplished by having trained people that go out and continue the process.
I'm ready to sign up as your graduate student. So, well, Anne, thank you very much. I really appreciate you giving us a bird's eye view of you, your history, and your lab, and everything that's going on. So thanks very much again.
Thank you.